Oh, we're back. All right. So we were chattering away with we have Fred Brown once again, our epidemiologist, who has joined us, and he most weeks is with us. Occasionally has some family obligations or something, but he's here today. And I looked at his slide deck, and we're going to be talking about the various vaccine candidates in development. And most people, and I think Fred would agree, and until we get a vaccine and get it widely disseminated around the world, we're never really going to get our arms around COVID-19, are we?
That's right. Yeah, I think we're going to need to have a vaccine to come back to what they consider normalcy. So we take the vaccine, and you're able to go out and about without worrying about things, not wearing protective gear. And we're pretty good at controlling the virus with protective gear. I mean, it's pretty effective. It's just we don't wear enough of it, and it's hard to keep it going. It's just tiring, always wearing a mask and everything. So vaccines definitely will be helpful.
Yeah, I keep a couple in my car. I keep a couple by the front door. I mean, sometimes I forget to grab it on the way out. Even when I go over to the office here where I live, I can't go in without a mask. And sometimes I get all the way to the door and I go, forgot the mask, So yeah, I'll be happy to get past that. And of course, me being a big sports fan and a Michigan season's ticket holder, well, that's not happening this year. So we're waiting to see what's going to happen with basketball. Maybe the same scenario, just the players on the floor and no people in the stands. But anyway, I digress. So vaccines, take it away, Fred.
Vaccines. Why don't I show you some of the things I've been thinking about? And
Yeah.
I'm working, full disclosure, I am working with the government. We have a special operation called Operation Warp Speed, and I do help that group out quite a bit. And so I'll share with you what I can today, and then in 2 weeks I'll be able to share a lot more, because I'm going to get a big presentation together. And be able to share and get that released and be allowed to talk much more generally. This is all material that is basic immunology. So we're going to talk a little about basic immunology, and that'll kind of prepare us for the more advanced discussions we'll have in a couple of weeks when they allow me to talk in more. So let me see if are you guys seeing everything so far?
Yeah.
Perfect. So let me go to the slide view so we don't get confused. And
if you could take it full screen, there we go. Yeah, we can read it better that way. So good.
So this is the landscape. We have 170 candidates. Typically for a disease at any given time, we'll have about 9. And why do you think we have so many candidates, Mike? Why do you think?
Well, I think strangely enough, they're all cooperating to begin with. And of course, it's a worldwide pandemic. And so everybody and his brother is trying to get involved in finding a way to solve the problem, right?
That's part of it. And the other part of it is we don't know what the heck we're doing.
I guess that really boils it down, doesn't it?
Unfortunately, I wish we could say, it's this way and we're going to figure this out and this is the way we're going to go. But there are a lot of different approaches because we just don't know enough about this virus. It's only been around for a little while, as you know, since January 11th, basically when we characterized it in the DNA. And so we have all these different approaches. What's interesting is we have 170 plus candidates and about 350 exploratory approaches, but they all boil down to 6 to, depending how you define the science, 6 to 8 approaches. And so these are the 6 approaches that this was published in Nature that people kind of talk about, the 5 or 6 approaches that we talk about. And each of them have kind of positive and negatives. So the first one you hear a lot about is this recombinant viral vector vaccine. And that's the one that J&J and AstraZeneca are doing, and they have basically what you do is you take a little vector that you already have, adenovirus or something that causes colds or something that causes a disease in chimpanzees, as in the case of AstraZeneca, and you take out that DNA of that virus, and you put in something that's going to create an antigen for COVID. So something that's a COVID protein instead of a adenovirus in this case, or in the other areas. And there's some good things about this. The first is that we know quite a bit about the virus vectors. There's some successful platforms out there for rabies and others. We can deliver it through the mucosa, and we think that's going to be important for a respiratory virus, being able to sniff the virus into the vaccine into you rather than inject it. We think could have some beneficial But of course, there's some risk in terms of the vector you choose. If you choose the wrong vector, it won't get in necessarily as efficiently as it should. It could be attacked by the body itself. So there's some challenges about using this vector, but we're pretty far along in the J&J opportunity. We should have a J&J vaccine, if it's successful, out by the end of this year.
Really?
Yeah. The nucleic acid base is, of course, the fastest one, and that's the Moderna and Pfizer ones. And Moderna and Pfizer could actually be out in November with an emergency use. Now, these are all assuming emergency use authorizations. And the problem is it's very unproven. We don't have any vaccines that have ever been discovered or produced this way before, and so we're sort of rolling the dice here. But you can see how fast it is. The vast majority of clinical development at phase 3 level is in the blue color, and you can see how many there are, how many candidates there are just in using this piece. What's interesting is, if Moderna works, it's likely that Pfizer will work. It's likely that if Pfizer works, all the other ones like this will likely work. And so, but at the same time, if it fails, it's likely that the others will fail as well. So it sounds like we got a lot of candidates, but the truth is that if one of these whole platform technology fails early, chances are the other ones are going to have trouble too. So we don't have as much diversity as necessarily we think we do, having 170 different candidates, if that makes sense. Because everyone has the same spike protein basically in this little envelope, and they're trying to push it in using a couple different vectors, a couple different lipid platforms, and that's all we got. Then we have the inactivated virus. The inactivated virus is one of our go-to ways of dealing with viral diseases. And basically, what you do is you take the virus as it is whole, you culture it up, and then you add chemicals to it, or you UV light it, or you heat it, and that changes the it actually inactivates, it kills the proteins. The problem is that sometimes the proteins, when they start to denature, start to change configuration as well. And so sometimes the antibodies that you're developing to the inactivated virus aren't necessarily the ones you're going to need against the real virus, because by definition it's been inactivated. On the other hand, it's very safe because the virus has been killed, but there is that chance of not having very good efficacy. The strongest response actually comes out from live virus. What we do there is we actually take the virus in its live state. We don't kill it, we keep it alive, but we weaken it. And we weaken it so much that it mixes with that it's not virulent anymore. And then we create antibodies to that weakened virus. Now the risk is, especially in coronavirus, that it sort of sees a wild type, what they call a wild type coronavirus or original coronavirus out there. It mates with that, and it conjugates with that with the wild type, and all of a sudden you got real virus again. And that's a real risk with coronaviruses, and it's called is a kind of reversion of the virulence back to where it was originally back to. That it does create the strongest immune response, but it also has the highest level of adverse events. So there's, again, positive and negatives. The final area is actually the protein subunit, and here this is the safest one. This is the one we're probably going to have to give to people who are immunocompromised, people who are very ill, because it actually just takes small pieces of the virus, you break it all apart, and then you take little pieces of the virus and you just inject that in, rather than trying to get something actively into the cells or trying to use the cultured virus. So in the United States, when the United States decided to do this work, they decided they weren't going to allow any culturing of the virus. So we don't have any virus, thing going against with live attenuated or inactivated virus. Everything we're doing is in the nucleic acid, the recombinant, and the protein subunit. That's all we're working on in the US. So that kind of gives you a sense of all the different areas that you could work on. And now here comes the immunology part of this. Basically, if you look at what's happening with the virus, it's two things. The first thing is how much of an immune response your body, how much of an immune response and how fast your body is able to recognize the virus. And that's the left-hand axis here, broad relative immunogenicity, right? So on this little score, if you've got broad immunogenicity, means you recognize it fast, it doesn't mutate much, and the protective duration of the primary immune response is pretty strong. And so as you go up that axis from low to high, it gets better and better for us. But what the virus does is it moves pretty quick. and it has what they call an incubation period. So the faster the virus is able to get itself established versus your immune system, it can then get established, and it won't matter. Your immune system is too slow, won't have the high ground, and won't be able to defend your body. So that's what's happening. It's basically your immune response against how long it takes the virus to get established. And if you can beat, if your body can beat the virus getting established, then you win the war. If you can't beat it, then the virus replicates and you spread it. And so you can see that generally we have very good, what the vaccines do is it gives your body a boost. It basically, gives you a head start in the race to react to the attack of the virus. And you can see that basically here, the squares indicate where we have successful vaccine. You can see it's all in the areas where it's taking the virus pretty long to get established and where you have a pretty high immune response. And you can see the flu's back down here where it goes so quick that we don't have a chance to get ourselves, go after it. And that's why we only have partially effective vaccines. HIV and AIDS viruses change so fast, we don't get a chance to really establish themselves against them. And then you got a whole bunch of viruses here, and you can see where SARS is. And unfortunately, SARS is very much 80% like the COVID that we're trying to fight. So here you can get a sense of where all the coronaviruses are, right? And we have we have 6 COVID viruses. Coronaviruses. 4 are colds, and we've never been able to create a vaccine against colds. And there's MERS and SARS. We still don't have a vaccine against them. And now we have the SARS-CoV-2. And you can see that it's right here in the square where we're right on the edge of our capabilities for a vaccine. So we're not sure whether we're going to be able to get a good vaccine here. At least we're not way out here with malaria and HIV, but it sure would be nice to be up here, where we have mumps and measles and so on. So we've never developed, the result of this is we've never developed anything against coronavirus on a vaccine basis. The vaccine immune response has been weak to date. And all we really know so far is what we've done with about 1,000 tests of humans and some macaque monkeys. And the problem with our immune response is it tends to be lower in the lungs as a result of the vaccine, not higher in the lungs. That's what we're thinking about inhaled vaccine eventually. So we might get a partially effective vaccine, but herd immunity is unlikely with these initial vaccines. And worst of all, the vulnerable patients may not be very responsive to these vaccines. So that sort of gives you a sense of the general immunology, what we're trying to look at here. And now let's see where we are. And so here, where the question is, gosh, we've got either, sadly, you can see we've got either very proven scalability in the vaccines or unproven scalability. And maybe next week or the week after, we can talk a little bit about what I'm doing with Operation Warp Speed to improve the scale, distribution, and administration of the vaccine in the future, because that's going to be a big challenge. But basically, you can see that the conclusion that we came to, this is back in January, was cocktail scenario. The fastest route to some control may be convalescent plasma and monoclonal antibodies, eventually creating a bridge to getting to a vaccine. And that's what we were talking about back in January. So what does that look like? Well, here's what it looks like. Basically, this is the immune response, right? So you've got 2 different kinds of immune responses. The first one is just antibodies that are circulating in your body normally, and that's the humoral response. And sometimes they'll latch up against the COVID virus, and it'll recognize it, and it'll kill it just because it sees it as a foreign object. Now, what we've heard about is in convalescent plasma and monoclonal antibody from President Trump was that basically this boosts the antibodies that are going around your body normally. So all of a sudden you get this special boost from, initially the convalescent plasma is from other people who've been infected. So you take their blood, you spin it down, you take their plasma, and then you boost your own plasma levels of the antibody because you're getting theirs. And that, of course, means that you're going to be able to fight and react better when you get the disease. So we use it in 2 ways. One is you're a healthcare worker and you're going to get exposed every day. You might want to use this, what they call prophylactically. You might want to use it in advance before you even get into a bad situation with a patient. So you're ready. And this effect usually lasts between 3 months and a year. And again, it drops. Over time it starts to decline, so it wanes because you're slowly you're getting diluted. The antibodies get diluted and pulled out of the system. The monoclonal antibodies actually are stuff that we produce. So we actually look, and fortunately in the plasma, you're getting everything. You're getting stuff that works, stuff that doesn't work, stuff that's very directed, stuff that isn't directed. And what we're looking for actually is what they call neutralizing antibodies in that plasma. We isolate those antibodies, and then we synthesize just those special neutralizing antibodies. And basically what you're getting is what I call an immune system in the vial. So you put it in and you get yourself boosted up in the immune system, and it's a bridge to the vaccine. But the thing about this is it doesn't stimulate your body to actually create its own defenses. And that's what you really want. And that's the difference between the antibodies just getting injected, someone else's antibodies or synthetic antibodies, and a vaccine. Because the vaccine, you actually have what they call cell-mediated response. You actually have the antigen that's being presented to the body. The body then turns on its machinery to create even more of this. And suddenly you're getting a big permanent response because you constantly have the machine turned on to producing antibodies instead of antibodies that are injected externally and then slowly decline over time. So that's the big difference between a vaccine and a monoclonal. That's why we talked about the fact that a monoclonal antibody might be a bridge to a vaccine because they're both involving the immune system, but differently in each way. So the issue we've got with monoclonal antibodies, and I told you yesterday that I thought the trick would be to go to your doctor for a loved one in the hospital, if you're in the hospital yourself, and ask for the Regeneron cocktail, or the Lilly cocktail, or the AstraZeneca cocktail. They're all under clinical trials right now, and see if you can get one. And a lot of our teaching hospitals in Michigan do have trials that are ongoing and participating in some of these trials. The issue is, sadly, we don't have enough demand. We don't have enough supply. So I did all the math after we talked, because that was one of your questions, I remember. And basically our demand for this, if we look at what we're anticipating demand to be for sick individuals in a hospital in the November, December timeframe, we think we're going to need about 1 million doses a month to take care of all the people and all the healthcare workers who are likely to be frontline. Mm-hmm. Unfortunately, we only have about 100,000 a month. And we'll slowly scale that up. But these are biological systems. You have to ferment them. You've got to purify them. Then you got to freeze them up. And then you got to send them out. So it takes a long time to build up the capacity. And so you can see that we bought about 1.5 million doses total, as the United States government did, for several $1 billion, 2.1 billion, I believe we spent. And that's only going to be about enough to supply about 1.5 months of demand. And that's all we've contracted. So we need a lot more of this. And I don't know if you remember, the reason I put this slide up that I showed you back in kind of March to April timeframe, Mike, was because we said, watch for this convalescent plasma opportunity, monoclonal antibody cocktail opportunity. We were saying that back, January, February, March. I don't know if you remember, but March, April, when we had a chance to really scale this baby up, everyone got sidetracked by hydroxychloroquine. And so instead of focusing on something that might have worked, it's easy to say that in hindsight now, we were pretty sure as immunologists it was going to happen. We focused on hydroxychloroquine. 70% of all of our resources went to that for the next 3 months, and we didn't scale up our capacity for this area that does look like it's going to work. Now, it's still under investigation, so we're still trying to find out whether it's really going to work or not. But that was obviously a misstep, and we should have been. Right now, we only have about 1/10 the amount of capacity we actually need, sadly. But hopefully we'll start to move that. There's still a lot of question about whether this monoclonal antibody stuff works, frankly. The statistical significance, we're talking about a 4% shift overall in death rates, and long-term stay hospital shifts. And that's just barely statistical significance in the case of Eli Lilly, slightly more statistically significant in the case of Regeneron. And then the question is, which cocktails are right? How many monoclonal antibodies do we really need? And that all those things still are still being worked out in clinical trials. So
let me ask you now, I'm not an epidemiologist. You are obviously. Going forward, as we develop all these variations, does the ethnicity in any way enter into it, where one race might be able to accommodate one type of vaccine and another? I have no idea. Or is it one takes care of everybody?
So that's a great question, and I think what's going to happen is we're going to have to stratify the population, unlikely by ethnicity, more by your health system, your health situation and age. I think there's going to be certain drugs. If you're older, that will be better. I didn't bring my watch. I don't know how are we doing for time.
Let me look here. We got about 10 minutes to go.
Perfect. So let me talk a little bit about your personal selection, because that's going to be toward the end. I will I'll talk about kind of selecting what thing is probably right for you depending on your stratification. We don't think it's going to be based on ethnicity, though. We think it's much more likely going to be based on your behavior, your exposure levels, and of course, sadly, a lot of minorities and people of color are at the higher risk groups because they're employed doing frontline work, a much higher percentage than whites are, and they also are working in more densely populated areas frequently in the cities and working in tighter quarters, both at home and on the job. So we do see if you're black in the country, you've got a 3.6 times higher rate of death than if you're white. With this disease. So we do want to give them preferential access to the vaccine when it becomes available, because it should reduce death significantly if successful. But you can see I've circled the cytotoxic T cells and the memory helper T cells. The reason I did this is some of the vaccines that are being tested are only looking at memory helper T cells. And I don't think that's going to be sufficient. I think they're going to need to have both the cytotoxic T cells and the memory helper T cells at high levels of titer in order to be able to control the virus. And you can start to ask these questions of the media and the doctors. How much of a response are you getting? What your titer levels are of helper T, cytotoxic T. Those are the kind of questions we're asking all the time in order to decide which vaccine is going to be better. Likely better or worse. And I've already gone through the idea that we've got two different ways of doing this, one with whole virus, the other with pieces of the virus. And that's how we create these antigens that go ahead and then fight permanently. So that's the story about transitioning from antibodies to vaccines. And I think that's going to happen. I think the transition, we will have the monoclonals out probably, November, December timeframe. EUA probably about the same rate. Emergency use authorized at about the same rate. The vaccine will also be available. And I think we'll be using all these different tools and our masks and all of our social distancing in concert to control the vaccine. I don't think the vaccine is going to be completely sanitizing, because we won't have enough of an immune response in the first rounds. We'll find out more as time goes on. And not everyone will be eligible or even want to have the vaccine because of side effects and the chance of making things worse.
Well, and the other thing is, in some cases, it may be multiple doses, you were saying, where you get 1 round, then 2 rounds, maybe 3 rounds. And just in the United States alone, that's a billion doses if everyone was going to take it, right?
That's right. Yeah, we think we're going to need kind of at least 500 million. If not more, 1 billion doses, just for the U.S. And you can see a great point. Here are the different ways of going after the vaccine, the 5 vaccine types, and you can see everything's related to the spike protein, because that's the conferred protein. And there's a few other things attached to the spike protein that different companies are focusing on versus others. Basically, it's about whether we can get the spike protein, which is the one that sticks into the ACE2 receptor and allows the vaccine to inject itself into the cell and cause disease. And you can see basically there are kind of 3 or 4 areas that we look at. And you mentioned dosing, Mike. When you look at so this is the live attenuated virus. Here you only need 1 dose. You take the live virus and it's there and it's a big response, but there's a big safety issue. Probably only about only 80% of the people can take this kind of a vaccine, but it is our go-to production case. It is our go-to technology typically in the long term. And I think that it'll ultimately be a live attenuated virus that'll probably control the vaccine, the virus the best in time. You can see that the attenuated virus, that there you need to have 2, maybe 3, and if you're older, maybe even 4 doses. And that's a lot of vaccine to take, right? The same thing's true about the DNA. We think we're going to need 2+ doses. Again, probably maybe even 3 or 4 if you're over 65. Same thing true about the viral vector. You'll need to have just 1 dose of the viral vector for Johnson & Johnson, 2 for AstraZeneca, and 2+ doses for the subunit spike protein. You can get a sense of the trade-off we're having between immune response and safety, right? The higher the immune response is, chances are you might have also a little bit more safety issues, and especially in the elderly and the high risk populations. And then you can see also in the leadership areas, the US is actually, I think, in second place. The country that's in first place is China. And the reason I say this is because they've got, they have inactivated virus and they're already injecting into their military. They've got the mDNRA. They have two different approaches here. They have the access to the Pfizer vaccine and their own, and they have the spike protein viral protein vector. That's the one that Russia used in Sputnik, by the way. If that's the adeno5 virus, I don't think it's going to be terribly effective, and I wouldn't take a Chinese or a certainly not a Russian virus. And you can see at the end here, we've got something that the immune response is quite low, but it's very, very safe. And that's the spike protein. Those take a lot longer to develop. And so you see that Novavax, Sanofi, GSK, those companies are about 6, 7, 8 months behind because they had to actually synthesize all those proteins first, put them into a particle, and then start moving, versus the others that just had to pull out the RNA and put it into a lipid system. And you can see that, as you pointed out, Mike, it's going to be different types of, you're going to have to pick and choose the vaccine based on a couple different things, right? First is your dosing, what kind of additives are in there, more additives you got. Some of these vaccines have literally 1,000 different pieces of content in them beyond just the antigen. Yeah, so you can get a lot of allergic reaction, and you don't want to have, you have to go back 2, 3, 4 times and time this thing. Especially if you're in a compromised situation. The route of admission can be administration can be very important. And then, of course, prior health history is going to be important, whether you have diabetes, whether you're predisposed, whether you have cancer. All those things are very important to decide which of these you want to take. The genetics, male, female, especially here, allergies and illness predispositions. If you're predisposed to some illnesses, and you know that, you really want to avoid the vaccine, the COVID at all costs, then you're going to want to take something that's a little more powerful, versus if you're just trying to reduce the symptoms of getting of COVID, then you might want to take something a little bit weaker. And then, of course, our lifestyle, right? What our diet, exercise, stress, smoking, whether we're able to keep these things, the microbiome, that all affects our responses. So anyway, those are some of the things to think about. And then personally,
we've got 1 minute left, by the way, Fred.
1 minute. So here are the things to think about. I would go to your doctor, and I'd be watching the signs, and I'd ask them, first, was your population cohort in the phase 3 trial? If it was, that's great. Look at what happened to that population cohort. What type of technology is this care, and what kind of safety profile and efficacy profile does that technology as a whole have? Take a look and make sure you understand all the safety and efficacy pieces of a virus. And for effectiveness, choose the one that worked in your age group and population type. If it's 1 or 2 doses, as you point out, Michael, people over 60 may have to take even more than that. Your overall health, and whether that's EU or FDA approved, make sure it's FDA approved. And your personal situation, if you think you're at higher risk, then you probably want to take this drug, the vaccine, earlier. Finally, if your community is likely to outbreak, I would take the vaccine earlier as well. But do get your vaccine, your flu vaccine now. So that's sort of what I have to say. What do you think?
It's very interesting, and we'll get your slide deck up there too along with it. If you want to find out more, all of Fred's various videos are at fredbrown.com, and he works with lots of different governments around the world and lots of different states. So he's our resident expert, and we always appreciate all the information that he gives us every week.
Additional four-year students love Lawrence Technological University's thriving campus life. But LTU has always met non-traditional students' needs too. Lawrence Tech offers over 100 degree and certificate programs that can get adult students started or back on track. And most of our classes are conveniently offered evenings at our beautiful Southfield campus or online, so you can balance your social, family, and work life even while you power up your career. Lawrence Tech, where Blue Devils dare.